Good morning students, 12th standard biobotany class. So last two class we see the vegetative reproduction, asexual reproduction, but in this class we will see the sexual reproduction in plants. So why we call it sexual reproduction? The involvement or fusion of a gametes to produce their young one is called sexual reproduction. So all angiospermic plants are also involved in a sexual reproduction. So last class we see the parts of flower. So name the parts of the flower. So there are four whorls of the flower: calyx, corolla, androecium and gynoecium. So already all of you know that in 10th standard what is a calyx, what is a corolla, androecium and gynoecium. So now we will see the essential parts of the flower. The androecium and gynoecium are called essential parts of the flower because it involves in the direct role of a reproduction. So what is androecium? Androecium is the male reproductive. So look this flower. So this part is called as an androecium. This yellow color portion is called as androecium. So this called is the male reproductive part of a flower. So it's made up of a stamen. It's made up of a stamen. Each stamen is divided into the tip region is a bulge-like portion is called as an anther, and attached portion is called as an or stalk-like portion is called as a filament. Then each stamen is producing the microspores or pollen grain. Microspores are pollen grain. So this pollen grain is also carried one flower to the another flower, that is for fertilization. So this process also involved in a plants, they are called as an sexual reproduction plants. So we will see the detailed process in a male reproductive part. So next topic is a development of anther. So at the beginning stage in the stamens, an anther is a homogeneous mass of a parenchymatous cell. Homogeneous mass of a parenchymatous cell. At the initial stage, the stamens are formed from a mass of a parenchymatous cell. So each one is called as a cell. It looks like there is no differentiation in this cell. Later it becomes a four-lobed. Then below this lobe, there is a small cells undergo division. So this cell is called as an hypodermal cell. These cells are called as a hypodermal cells, undergo division, gives rise to archisporial cell. And then outer wall is called as a parietal cell. So these cells are also produced from this hypodermal cell. Then each archisporial cells undergo periclinal division. What is periclinal division? This is an cell. So parallel to the surface, when you divide this like, this is called as a periclinal division. So this gives rise to outer and inner cell. So the archisporial cell undergo periclinal division to form an outer parietal cell and inner sporogenous cells are also formed. So this region is called as outer region that gives rise to wall layer of the anther, and inner cell is archisporial cells developed undergo again divide periclinal and anticlinal. The archisporial cells divide again, this calls a periclinal, again divide anticlinal. So division is called as anticlinal, and again this like, and again. So this calls a repeated anticlinal and periclinal division, that is gives rise to 2 to 5 layers of anther wall. So the primary sporogenous cells gives rise to sporogenous tissue. So initially a mass of a parenchymatous cell is differentiated into outer parietal wall and inner archisporial cell. This archisporial cell again divided into anticlinal and periclinal division and gives rise to sporogenous tissue. So sporogenous tissue is the formation of a microspore. So how the microsporogenous tissue developed? So the microsporogenous tissue is diploid in nature. So this cell is we are called as a microspore mother cell. This microspore mother cell undergo meiosis cell division. That is gives rise to microsporogenous tissue. So this cell is the inner cell, this cell is microsporogenous tissues. So these microsporogenous cells undergo divide and redivide by mitosis. Remember, the diploid microspore mother cell undergo division is called meiosis. And then microsporogenous tissue divide and redivide gives rise to a form of mass of a sporogenous tissue by mitosis. Sometime asking one more question. The diploid spore mother cell gives rise to meiosis division. The microsporogenous tissue given. And then this microsporogenous tissue is developed, gives rise to sporogenous tissue by mitosis cell division. So this mass of a sporogenous tissue functions as a spore detrod. This is called a spore detrod. So inside this sporogenous tissue undergo division and functions as a microspore detrod. This is called a tetra means a 4. Then these are the 3 cells, then back side is another cell. So this microspore detrod is a haploid, but microspore mother cell is a diploid nature, all of you remember. Then next one, in some plants, so these microspores detrods are also separated, but in some plant, all these microspores in the microsporangium remain held together to form a single structure. They are called as a pollinium. What's the name? Pollinium. In some plants, the microspores are also produced in the microsporangium, remained held together in this microsporangium. This is called as a pollinium. Example, calotropis. We say in Tamil, Irkambu. Then this pollinium are attached to a clamp. The pollinium is also attached to a clamp-like structure. This is called as a pollinium. It looks like our lungs. So attached to a clamp-like structure. Clamp-like structure is called as a corpusculum. What's the name? Corpusculum. So then filament or thread-like part arising from each pollinium is called as a retinaculum. What's the name? Retinaculum. So this whole structure polliniums looks like a inverted Y letter. So they are called as a translator. What's the name? This is called as a translator. So these are also what is a pollinium, corpusculum, retinaculum, the translator. Somebody asking 1 mark or 2 mark question. So thoroughly prepare this topic. So now we will see the transverse section of anther. That means TS means a transverse section of mature anther. So already told that each stamen consists of anther and a filament. Anther is a fertile region. So how this anther cut or in a transverse section. So what are the parts, what are the changes occur in the anther? We will see one by one. First one, anther is a bilobed. Bilobed means this like. This is a bilobed structure. So each lobe having 2 theca. 2 theca means a chamber. Theca means a chamber. This is called as a dithecous anther. Monothecous, trithecous, tetrathecous are there. So in this given the syllabus is called as a dithecous anther. A typical anther. Typical means an example of anther is a tetrasporangiate. Tetra means 1, 2, 3, 4. This is called a tetrasporangiate. So bilobed means a tetrasporangiate. So what are the parts in the mature anther. First one is called as a anther wall. So it consists of epidermis. It consists of epidermis, endothelium, middle layers, and tapitum. It's called as a tapitum. So this layer is called as a tapitum. So anther wall consists of epidermis, endothelium, middle layer, and tapitum. First one, epidermis. So this is called as the outermost layer of the anther wall. Outermost layer of the anther wall. It is made up of a single layer. It's made up of a single layer. It protects the anther. So that's a protective in function. So first layer is called as epidermis. It is made up of single layer. It's made up of a single layer. Single layer and it's protective in function. Next layer is called as endothelium. It is also a single layer, but cells are arranged in a radially elongated cells found below the epidermis. So the inner tangential endothelial cells are divided, and sometimes it's called as also destroyed, or this called as disappear in this some plants. Next one, the inner tangential endothelial cells are made up of alpha cellulose. Sometimes it's a hygroscopic in nature because of during the dehiscence of the anther, it absorbs more water and helps for the dehiscence of anther. This is called as an hygroscopic in nature. Hygroscopic means absorbs more water. This is called as water loving. Endothelial cells are absent in some plants. Endothelium is absent in some plants. Example, aquatic plants. Aquatic means a water plant. And a clistogamous flower. Clistogamous flower means the flower is never open. They are also only in closed conditions. And some of the examples are called as extreme parasite. Do you know the parasite? So that is some of the plants are also depending upon the food for other living organisms called a parasite. So these are the extreme parasites. The endothelial cells are absent. The central part of the anther is lacking this thickening. This thickening means this endothelium thickening is also absent. So that portion we are called as a stomium. What's the name? Stomium. It helps in the dehiscence of anther during maturity. This called is lacking this thickening. This endothelial thickening is absent in this region. So this called is it helps to dehiscence to anther and releasing the pollen grains outside. Then third layer is called as middle layers. So this layer is called as middle layer. So these layers are also 2 to 3 layers of cells. 2 to 3 layers of cells next to the endothelium. They are ephemeral. Ephemeral means short-lived. So there is also, after the maturity, these cells are also crushed by the development of microspores. That's also destroyed. The next layer is called as tapitum. Tapitum is very important. Innermost layer of the anther. Innermost layer. So outermost means the epidermis. The innermost means called as tapitum layer. It is dual in nature. Dual means two purpose. So this layer, this tapitum is also partly developed from the primary parietal cell, and partly developed from the connective. So the tapitum is also partly derived from the primary parietal cell and partly from the connective cell. It is said to be a dual in nature. So the cells of the tapitum may be uninuclear, it's a single cell, sometimes many cells, and sometimes it becomes a polyploid. This cell is more than 6 or 7 nuclei. There is no cross wall occurred, it will become a polyploid. So what is the contribution of the tapitum? It's also the helps in a pollen wall material. material called sporopollenin pollen kit. Sporopollenin and pollen kit, and a tryphin and a number of proteins that control the incompatible reaction. Suppose example, a large number of plants releasing the pollen grain. Suppose these hibiscus pollen grains also falls on the another hibiscus flower. This is compatible. If suppose in this area you have a large number of flower dehiscence at the same time, what happened? So so many pollen grains also falls on the stigma. It will not germinate on the stigmatic lobe. This is that helps in the pollen. This is incompatibility. This is ovvathanmin solluvaanga. And it also controls the fertility and sterility of the microspore. So it also controls the fertility and sterility of the microspores or pollen grain. That's a tapitum. Tapitum is very, very important for 3 more questions. So this is sexual reproduction is a very important for your examination point of view. For example, what is a stamen? What is a stamen? And a drawn label the structure of a mature anther. Drawn label structure of a mature anther. Write short notes on the tapitum and write the functions of a tapitum. What is a middle layer? So you have to prepare this topic, very important. So that's all.